Hi, welcome to Practically. We bring learning alive. Yesterday we had started one series known as Evolution from Micro to Macro, and today we are going to continue that series with again prokaryotes. But this time we are going to take the eubacteria, that means the true form of prokaryote known as bacteria. So let's begin our session. So yesterday we established that there were some things, some creatures in the water, correct? The water may not be purified because purified water usually kills all the organisms, but the tap water does contain some creatures, and those are bacteria. So today we are going to see bacteria. Bacteria come in many different shapes and forms, and they are almost everywhere around us, right from your hands to your face to your mouth, nose, in the gut, everywhere around you. Even the water that you drink may contain bacteria. The air that you breathe also has bacteria in it. And bacteria are only visible through the help of microscope, which we established yesterday. Microscope is an instrument which helps to microscope helps the magnification of bacteria. We can't see bacteria with the help of magnifying glass, but only with the help of microscope, be it simple microscope, compound microscope, or any higher kind of microscope. So in today's session, we are going to see some of the basic properties which are similar to the prokaryotes, known as, so you would like to know something about bacteria, right? So the properties start with shapes. As I have told and shown some images that there are many different shapes of bacteria. Hi chemistry mate easy. Hi Harsh. Bio, yes. So bacteria come in many shapes. Then they are found in different habitats. As I have told you, microorganisms or bacteria are present everywhere around you. Isn't it? Right in the air that we are breathing, the surface that we touch, or the chair where we sit. Everywhere bacteria are found. Therefore, they are present in different habitats. Then they can be either aerobic or anaerobic. This means that aerobic means they can survive in oxygen or they can survive without oxygen. Okay, so this is the primary fact. They can. What is difficult, Arsh? Hi, Shabana. Thank you, Preeti. So these are the facts. They can survive without oxygen also. And based on the staining procedure. Now, staining procedure is something which you will come to learn as you do your practicals, be it in class 8 or be it in class 9 or 10. So, staining is a procedure where you can color the organism with some chemical compounds. And based on the staining procedures, we divide them into gram positive and gram negative. We will study this in the later slide. Then, organisms can be good. You might have heard that organisms present in yogurt or dahi is useful for your gut flora. So organisms can be good, but they can be bad as well. How? Because bacteria can cause different types of infections, isn't it? As I was saying, based on the shape, bacteria are classified or categorized into, oh, Arsh, tomorrow is your birthday. Great. In advance, I am giving you happy birthday. So based on the shape, we can categorize as coccus. The round shape of bacteria, which is found in some of the bacteria, are known as coccus. Coccus is a big family. Then we have rod shape. Rod shape bacteria are known as bacillus. Okay. Whereas the combination of round plus a rod is known as coccobacillus. It is small rod but a little round in shape. Then we have fusiform bacillus, which have elongated shape and some tapering ends. Okay, so this is fusiform bacteria or bacillus. Then we have comma shape, right? So comma shape is known as vibrio, and vibrio is usually pathogenic. Pathogenic means it can cause diseases. Then some spiral shaped microorganisms or bacteria are also known as spirillum and spirochetes. Okay, so keep these terms and forms in mind for your future reference. So these are some of the properties of bacteria. Bacteria being a prokaryote have similar structure design. Isn't it? Yesterday we saw that prokaryotes have structure design where they have cell wall, they have cell membrane, they have cytoplasm, ribosomes and nucleus. They don't have nucleus, but they have nuclear material known as DNA. Okay, and DNA is present in the central part known as centroid. Isn't it? We heard this term, right? Nucleoid. So this is where the DNA is present in the central part. Okay, and based on staining procedure, again I have said, they are divided into gram positive and gram negative. See, this is how the staining procedure is done. In this case, we use two different types of stains. Crystal violet stain and we use saffron stain. These are chemicals. Okay, these are chemicals which specifically color some compounds or some structure present in the bacteria. So in this procedure, what we do is we take some small drop of bacteria onto a glass slide. So this is the glass slide. Okay, and we take small droplet of water which may contain organisms. Then we add crystal violet and wait for some time. Then we add iodine solution. This is known as mordant. Okay, mordant. Why? Because it fixes the crystal violet into the bacterial cell. This you will understand more in depth when you are doing your practicals. Once iodine is added, then you add ethyl alcohol to wash away the excess amount of stain that is present in the slides. And then we are staining with saffron. Now, crystal violet gives violet color. Okay, and saffron gives reddish pink color. So this is how organisms are stained. If your bacteria is gram-positive bacteria, it will take the crystal violet stain. Okay, and if your bacteria is gram-negative, it will take saffron stain. And we always require a microscope to observe this type of procedure. Okay, so keep this in mind. So what are the differences between gram-negative and gram-positive? Well, as I have said, they take up violet color, that means purple color appearance in the gram staining, and because of that they are known as gram-positive. Whereas pale reddish color or pink color appears after saffron stain. So these organisms are known as gram-negative. Then we have bacteria that have shape known as cocci. Okay, as I said, cocci is a round shape, isn't it? Spherical shape. So a cocci, in staphylococci, there are berry-like structure, that means grape-like structure. So these shape combined together are known as staphylococci. Whereas in streptococci, they are in strip form. They are in strip form, that means they are chain form. Therefore, they are known as streptococci. And some listeria species. Listeria is again some kind of bacteria which causes disease in human beings. Whereas in case of bacteria in gram-negative, we have enterococci. Again here shape is cocci, but it is entero, that means it is inside your body. Therefore, it is known as enterococci. Whereas salmonella, I think all of you might have heard this term salmonella. Arsh, are you there? If not, salmonella species causes different types of diseases, specifically known as poisoning. Salmonella causes food poisoning. Then we have Pseudomonas. Pseudomonas is usually present on your skin and it may cause you some skin diseases. They have in gram-positive thick peptidoglycan layer. Now peptidoglycan is a chemical, carbohydrate-based chemical, okay? And that is present only in bacteria, not in archaebacteria, okay? Keep that in mind. Whereas in gram-negative, we have thin peptidoglycan layer, okay? Then lipid membrane is absent. As I had told you, in archaebacteria, they have lipid membrane. But in case of bacteria, in gram-positive, we don't have lipid membrane. Instead, lipid membrane is present in gram-negative. Okay, they are present in gram-negative bacteria. Anticoic and lipoteichoic acids are specific markers, you can say, to distinguish the gram and gram positive and gram negative bacteria. These compounds are not present in archaea, which we studied yesterday. In archaea, these compounds are not present, and because of this, this is a distinguishing feature between bacteria and archaebacteria. Okay, so this is how the stain will look like. Okay, pink and purple. So again, this is how I was saying difference between gram positive and gram negative. Plasma membrane is present in gram positive. Periplasmic space is present in gram positive, as well as peptidoglycan. This is a distinguishing feature. Okay, this is a distinguishing feature between gram positive and gram negative. Okay, in negative, you can see there are multiple layers of membrane: plasma membrane, periplasmic space, peptidoglycan in small amount, and lipopolysaccharide and protein, that is lipid membrane, which is absent in gram positive. And this is what they can look like: purple color. You can see there is some bulging in the bacteria. Yeah, some bulging in the bacteria. That is nothing but spores. Spores, because bacteria can reproduce asexually. That means they can only reproduce asexually, I meant to say, by binary fission, fragmentation, etc. And if there are some conditions which are adverse for the growth of bacteria, adverse in the sense if they are not proper feasible conditions, then the bacteria will store themselves in the form of spores. That is their defense mechanism, and once the environment is good enough for them, they will again flourish, they will again multiply and grow. This is absent in archaebacteria. So now we should understand how bacteria are bad. Well, bacteria causes different types of diseases. Among them, skin disease is one part. Then they can cause dental caries to your tooth. Then they can spoil your food, isn't it? Spoil the food. Then cause dental problems. Cause skin infection. Okay. Or if you eat some bad food which is already spoiled, then you can have upset stomach, food poisoning, isn't it? So bacteria are bad. These are few examples, commonly known examples, but other than that, they can cause deadly infections also. Then you might think, what are the advantages? As I have told you, bacteria are also present in your gut, in your stomach, isn't it? So these bacteria help in the production of vitamin K, which is helpful, and it also helps in the digestion of food. Then in dairy products, right? Dairy products like yogurt or curd, you have probiotic bacteria. Probiotic bacteria like lactobacilli help in the digestion of food, which is very good. Then they can degrade the waste. If you have some, you can say wet waste, that means your food material or garbage which contains peels of your fruits and vegetables, those material are easily degraded by bacteria. Degradation of waste. Therefore, they are helped in bioremediation processes. Then they can also help in the oil spills. Now you might know that oil is obtained from oceans, isn't it? Big companies are mining the oil from ocean floor. So what happens is sometimes some of the oil is spilled, or you can say it is splashed in the ocean. So it pollutes the ocean. So what scientists are doing, they are using bacteria which can utilize the oil or which can consume the oil, and they are cleaning the ocean with the help of bacteria. So these are some of the advantages of bacteria. Now, what we are going to study in the next session, which will be on Friday, is we are going to studying about origin of life. So be with me on this episode on Friday. Okay. For more such interesting videos and content, do like our Practically Life channel and subscribe it. Okay. As well as download the Practically app from Play Store and App Store for more such interesting video content and educational things. Until the next time, bye bye from my side.